Okay, good evening. Let us start our talk on transgenic plants and GMO. These two topics I shall discuss in detail today. I welcome all the students who joined today and will be joining soon. Today is the Valentine's Day, so best wishes and love for all of you. And I wish you had great time during the election and let us hope for the best for the nation. Now, let me show you the slides and start my talk. First, tell me whether you can listen my talk clearly and how about the visibility of the slides? Is it clearly visible?
Yes, sir.
Yes, sir.
Okay, but it seems it is a PDF one. Let me find the uh, uh, PowerPoint slide. I guess this one, PowerPoint. Let me see whether it is PowerPoint. Yeah, fine. Okay, great. Yes, today we are talking on transgenic plant production and genetically modified organisms. I am talking from the Bangladesh Agricultural University campus. I'm here just 1 hour before I came here, and this is my alma mater. I graduated from this university in early 90s, and tomorrow we have an international symposium. But anyway, the guest house is peaceful, no problem. So, in our previous lecture, we discussed about the plant tissue culture, right?
Yes,
And
sir.
this is not been discussed before, right? Okay, the transgenic plant, is it already been discussed in the earlier lecture?
No, sir.
Okay, fine. Because I confused, I am offering another course also. So, transgenic plants, what is it? Let us get the concept, that is formal definition. Transgenic plants are genetically modified organisms that have been altered using genetic engineering techniques to contain one or more genes from other species. Transgenic, the word means genetically modified organism which is carrying uh, one or more genes from other species. That means if we modify the genome of a plant with uh, one or more gene from other species, then we can call it transgenic plants. And obviously, transgenic plants or transgenics or transgenic organisms are genetically modified organisms. So we can say all transgenic plants are GMO. So transgenic plants definition includes those plants which have one or more genes from another species introduced into their genome through genetic engineering process. Um, why transgenic plants are important? Uh, they are important because by genetic engineering, addition of new gene into the genome of an organism, we can improve the trait the of a plant. For example, enhanced trait, pest resistance or herbicide tolerance, improved yield or nutritional content. And we have a bright example of transgenic plant. which has been introduced in Bangladesh in 2014, is BT brinjal. BT brinjal is a brinjal, eggplant. We transformed this plant, genetically modified the genome of this plant by introducing a gene from a bacterium called Bacillus thuringiensis, BT. And the plant carries the BT gene is a transgenic plant, transgenic brinjal, GMO brinjal, which is resistant against certain insect pests, and specifically brinjal shoot and fruit borer insect. We have another uh, GMO plant, golden rice, which has been developed by Bangladesh Rice Research Institute. It carries a gene, beta carotene gene from the maize. Um, as a result, it can biosynthesize beta carotene in the grain, which is the precursor of vitamin A. That is vitamin A enriched rice. It is not yet been practically released in Bangladesh. It is now under the process of release by the uh, Department of Environment. If we modify the genome of a plant by a new gene, or deleting some gene, and if the outcome is higher nutritional content uh, in the edible portion, then it is called biofortification. Biofortification, another term in genetic engineering, uh, where we can improve the nutrient content. For example, in case of golden rice, vitamin A content in the rice, but a normal rice does not contain vitamin A. So biofortification is a process of genetic engineering. By that process, we can genetically modify a plant to with enriched or enhanced nutrient content. Contributions to the agriculture. Actually, genetic engineering or GMO has a robust contribution to agriculture, sustainability, and food security. Early genetic modification actually uh, technique was developed by several historic breakthroughs. I mentioned Paul Berg, who developed the recombinant DNA technology, or the work of Barbara McClintock, who discovered the transposable elements, that is, jumping gene, help us do the genetic engineering, those basic words. First commercial transgenic crop was flavor saver tomato. What was that? It was a new tomato variety which is uh, strong in shelf life. That means tomato easily after ripening, it is called climacteric fruit, easily, uh, spoil due to fungal as well as faster ripening. So in that case, flavor saver tomato, you after ripening, you can preserve for a bit longer time. So its shelf life is increased through the genetic engineering. Let us know more about the transgenic plants. Genetic engineering techniques are used to change the DNA of transgenic plants. This technique offers benefits like extended shelf life, I mentioned, flavor saver tomato, increased yield, I am repeating and reviewing again, improved quality, pest resistance. Pest means insect pest or disease resistance. Heat tolerance, cold tolerance, or drought tolerance, or tolerance to the salinity. So these resistance allowed to grow plant under a range of biotic and abiotic challenges. Biotic stress and abiotic stress. Biotic stress means uh, the living organisms like fungi, bacteria, nematode that cause diseases in plants and create the stress, they are called biotic stresses. That is, stressors are living organisms causing the disease. On the other hand, abiotic stresses are non-living, factors that impact or on the plant growth and survival. For example, heat, high temperature, severe cold, drought, that is scarcity of water, as well as salinity, and many more. So by using the uh, modification in the DNA, that is transgenic technology, we can uh, develop uh, ma- biotic and abiotic stress tolerant plant variety. Additionally, foreign proteins of commercial and medicinal value can be expressed in transgenic plants. What does it mean? It means by using genetic modification in the plant, we can, uh, you know, commercially produce important proteins or vaccine or some medicinal compounds by using the plant. If we introduce the gene responsible for synthesizing particular proteins useful in commercial purposes or medicinal compounds, even a vaccine. And in that case, the transgenic plants, cultivation of transgenic plants that can produce pharmaceutically important compound is called biofarming. Biofarming. Harming. Farming means it comes from the pharmacy. So, by using transgenic plants, you can make pharmaceutically important drug molecules. And this is another very useful use of transgenic or GMO technology in plant. Golden rice, as I mentioned, which makes beta carotene, is given the beta carotene gene. The most common source of beta carotene is carrots, which, when added to rice plants, begin producing beta carotene. So, rice plant then produce the beta carotene, and in the grain looks also golden. Golden rice is fortified with vitamin A to increase its nutritional value because beta carotene is one of the precursors of vitamin A, that is beta carotene. During digestion process we can get the vitamin A, and vitamin A is very very important vitamin uh, responsible for our eyesightness vision. So very very crucial vitamin. BT brinjal is an addition from the transgenic plant, and the first transgenic plant commercially being cultivated by the farmers. In that case, BT gene originates from the bacterium. Yes, Shubra, you can ask question.
স্যার দ্য গোল্ডেন রাইস এইটা কি স্যার আমরা যে বিন্নি চাল বা লাল চাল যেটাকে বলে ইজ দ্যাট দি ওয়ান?
No, lal chal, binni chal, they are the natural. They contain, different vitamins, but not the vitamin A. Golden rice, when it is cooked, even unhusked rice looks like, golden color. And due to the presence of beta carotene, I shall show you later on the image. The binni chal or other red or brown rice, they are naturally pigmented. Even we have the black rice also. They are not genetically modified. They are the natural one. Okay.
Okay.
Thank you.
Yes, sir.
So uh, the Bt gene originates from bacteria, can easily be inserted into the brinjal plant, as we did Bangladesh Agricultural Research Institute in collaboration with a company, a Mahiku company. They modified 4 to 5 brinjal variety using the Cry1 gene. Cry1 gene is called chrysine. There are several chrysine produced by the Bacillus thuringiensis, which is important for producing the toxin. Okay, it is the brinjal shoot and fruit borer is a powerful insect that can destroy the fruit and shoot of the brinjal, but due to the uh, genetic engineering, the genetically engineered or transgenic bt brinjal can produce its own toxin. No need to apply any insecticide, because to protect the brinjal plant, farmers sometimes use everyday insecticide, which is hazardous for human health, because even before the one day before the harvest, they use toxin, that is insecticide. But in case of BT brinjal, no need to apply the insecticide. What happened in this case? Cry1 gene, when expressed in the brinjal, it produces Cry1 SE protein. And this protein is toxic for the insect, but not toxic for the human, because it requires, it requires the uh, alkaline environment, that is pH more than 7, to be a toxin. In case of insect gut, uh, alkaline environment exists, but in case of human and other animals, our guts are acidic. So in acidic condition, this endotoxin never be toxic. So this is why we can safely eat the BT brinjal, but for the insect, it is hazardous, and this is why insect is do not insect does not infect the or infest the BT brinjal. Similarly, Bangladesh also introduced another GM plant. It is called BT cotton. Same principle, BT gene introduced into the genome of cotton plants. And that cotton plant actually suffers from uh, cotton bollworm, and this is a dangerous lepidopteran insect, destroy the cotton yield. So in that case, bt toxin can protect the bt cotton from the devastation. Development of transgenic. How can we develop the transgenic? There are several methods, but two major methods we are uh, popularly using. One is called biolistic uh, method, that is particle gun method. It is also known as particle gun method. Another one is agrobacterium tumefaciens mediated transformation method, um, and they are popularly being used. Biolistic method, in this case, we use a gene gun for delivering the DNA, that is your target gene, into the genome of your target plant cell. And by using DNA or RNA or proteins, we can introduce into the target cells uh, by using the gene gun. I shall show you image later on. But before using gene gun, we need to coat the gene or biomolecules, macromolecules with gold or tungsten, gold nano gold particle. We can coat the DNA, a small particle of DNA, that is gene, by using the gold or tungsten. And they are then introduced into the cell with very high velocity and pressure using an electric pulse or pressure in helium pulse. Such technique involved with the delivery of micro projectile is referred as a biolistic, that is biolistic gene gun. The major limitation of this technique is that the DNA is randomly introduced into the target cell. So you cannot precisely place the DNA in the specific location. Here is the image. Our left hand side, you can see the biolistic gene gun machine. And here there is a, the main gun point, gas acceleration tube, gene gun barrel. So what we can do, we can place the cells, especially we use the uh, protoplast, that is plant cell, when the cell wall is removed by chemical and other enzymatic process, then it is easy to, incorporate the uh, gene into the cell, because cell wall is a protecting agent. So first we remove the cell wall and make the protoplast and take in the plate. And then here you can see gene, the yellow one coated with the gold nanoparticle, and then you press. And due to high velocity, some of the particles can enter into the cell and even into the DNA and integrate your gene into the genome. But it is a physical process, and after that you cultivate these cells and to get through the tissue culture process and get the seedling. Some of the seedling you will find carry your introduced gene, and through the selection and screening you can get the genetically transformed plant. So this is one of the process worldwide people use. Another process is uh, transgenic plant development, is agrobacterium mediated transformation method. And this method is more popular than the previous one. Here in our right hand side, you can see a plant, infected by agrobacterium tumefaciens bacteria, developed uh, tumor-like structure. It is called crown gall or gall. That is like tumor, that is profusely grown cells. Scientists, especially plant pathologists, they discovered that this disease is caused by Agrobacterium tumefaciens, and this is a unique disease that is development of cancer in the plant. And how does it occurs? To understand the mechanism, scientists confirm that Agrobacterium tumefaciens carries TI plasmid, that is tumor-inducing plasmid. Bacterial DNA carries uh, bacterial DNA present in the cell in two forms. One is nuclear DNA, chromosomal DNA, another is plasmid. Plasmid is circular one, which is mobile DNA, can go out from the cell and can in. So uh, in case of Agrobacterium tumefaciens, the plasmid DNA contains TI plasmid, tumor-inducing plasmid. What is it? it can inject bacterial DNA into the genome of the plant. And this is one kind of, terrific war against the plant, even engineering the plant. So when the mechanism of agrobacterium and the plant was discovered, then researchers thought, is it possible to use this agrobacterium tumefaciens, which carries TI plasmid, as a tool for genetic engineering? And it happened. The agrobacterium approach is the following technique for creating genetically modified plants. Agrobacterium tumefaciens, a soil-dwelling bacteria, is used in this process. And commercially, you can buy the Agrobacterium tumefaciens, a virulent, isolate, and it is very quite expensive. So we can use this one. It has the capacity to spread a piece of its DNA into the plant cell. Through a tumor-inducing plasmid, the DNA fragment that infects the plant gets incorporated into a plant chromosome, that is TI plasmid. The plant's cellular machinery may be controlled by the TI plasmid, which can then exploit it to produce numerous copies of its own bacterial DNA. Very interesting. A sizable circular DNA particle called TI plasmid replicates separately from the bacterial chromosome. You can replicate and make too many copies, and you can insert your desired gene into the TI plasmid. And this plasmid is significant because it has areas of transfer DNA, tDNA, where a researcher can introduce a gene that can be transferred to a plant cell using the method called floral dip. A floral dip entails dipping flowering plants into an agrobacterium solution that contains the desired gene and then harvesting the transgenic seeds, very interesting, that is modified plant. I can show you here the two methods in an illustration, very simplified illustration. Okay. Here you can see what you need to develop a transgenic or GMO plant. First, you need to identify a gene which you want to introduce into the genome of a plant. So, for example, BT gene. So, this is the gene identified and isolated. You have to know its sequence, ATGC sequence. So, what you need to do first, you need to insert this DNA in the plasmid, that is circular DNA you harvested from Agrobacterium tumefaciens, because Agrobacterium tumefaciens or all bacteria carries chromosomal DNA and plasmid DNA. So you can easily get the plasmid DNA from the bacteria and cut the plasmid DNA by using restriction enzyme and then insert your DNA. And this, recombinant DNA, then you grow with the bacteria, and then as it is a mobile DNA, it can easily enter into the bacterial cell. That means that Agrobacterium tumefaciens carries its own chromosomal DNA and plasmid DNA with your desired gene. And then you do the floral dip method. That is, you cultivate this recombinant DNA carrying Agrobacterium tumefaciens with the plant cell in the same plate. As Agrobacterium tumefaciens has TI plasmid, which can insert its own DNA into the plant cell. By chance, the DNA you inserted can also transfer into the genome of the your plant cell. And in that case, you grow thousands of plant cells and thousands of bacteria together. And after that, you separate the cells and then do the tissue culture. And after tissue culture, you can find uh, some of the cells carry your uh, desired DNA, and then you cultivate and grow the plant. And this is the transgenic plant carrying your DNA. Actually, all the plants you grow through the tissue culture may obviously not carry the transgene. So, in that case, when you get the seedling, you need to check whether it carries the transgene or not. How can you do it? For example, if you introduce or develop a GM plant by introducing a salinity tolerant gene, then this plant will easily grow in saline medium, that is soil or water carrying salinity. But other plants do not grow. In that case, you can easily identify the salinity tolerant plant, but it is a phenotypic identification. What you can do the PCR and amplify the DNA and get the amplify the DNA, especially gene you introduced by using gene specific primer. And by PCR, you can easily identify which plants carry the transgene, or you can do the DNA sequencing. So this is the simplified process of the development of transgenic plant or GMO using agrobacterium mediated transformation. On the other hand, we can use the gene gun. In case of gene gun, before you use the gene gun, you replicate this gene, your desired gene. How can you do it? You can easily make copies by using PCR. Okay, so you can make thousands of copies. After that, this gene can be coated with the gold nanoparticles so that gene is protected. And by using this gene gun, you shoot on the protoplast, that is wall-less cells. And by force, some of the particles can enter into the uh, uh, genome of the plant cell. And by using the same process, you can through tissue culture, you can get the plant and screen. In that case, gene gun or the agrobacterium mediated transformation, most important is to check and screen and find the plant which carries the your desired gene. Sometimes the desired plant you will get a few percent. Most of them do not carry the transgene. So you need only one or two, and then you multiply and get the copies and see when you grow in the natural environment whether it gives the desired trait or not. Trait means the phenotypic or properties, that is salinity tolerant or insect resistance or even vitamin A enriched product or like that. So I would like to stop here because it will be included in your exam. Do you have crystal clear uh, um, understanding uh, on this slide? And if you have any question, you can ask me now. Yes, Mahmuda Haq.
Sir, can you go to the previous slide?
Yes, I can go to the previous slide.
Can you please explain me the floral dip once over again? I did
Yeah,
not get it
floral dip.
properly.
Floral dip means when you introduce your desired DNA into the DNA of plasmid DNA of the agrobacterium, and then you get the agrobacterium carrying your DNA, and then you grow the plant cells with the bacterium, as it is called floral dip. That is bacterial suspension. You add the plant cells, or you take the plant cells and you add the uh, bacteria containing the uh, gene that you inserted into the plasmid, and then grow both. And it is called floral dip. For example, you bring the flower sticks and put into the solution containing the minerals, and you put the stick, cut, and so that it can get the nutrition. That is in here. It means floral dip means you added plant cells, or you added bacterial cells on the other cells, that is bacterial cells with the deep into the plant cell, or plant cell deep into the bacterial cell, so that they can get in close contact and allow bacteria, agrobacterium tumefaciens cells, to transfer that its DNA, also carrying your desired DNA into the plant cell. So it is popularly called floral dip method, dipping flowering plants into agrobacterium solution or like that. So we usually use flowering plants for genetic transformation. Thank you. Any more question? Anyone? Just unmute yourself and ask the question.
Sir, after we have dipped the stems in the floral dip, okay, the transformation will happen. Like it will form the tumor formation will still happen, or it's like because it said that will collect the transgenic seeds or something. So
No,
will
no,
it
no
form
tumor,
tumors
no
or
tumor formation. Actually,
okay?
you use the plant cells and bacterial cells and allow them to get in touch. So uh, bacteria transfer DNA, because bacteria has tDNA and Ti plasmid, tumor inducing plasmid. So bacteria from its Ti plasmid, it can transfer DNA into the plant cell. So up to that, the job is done, floral dipping. And then you remove the, uh, plant cells from the mixture. You just kill the bacteria and then allow plant cells to into the tissue culture medium to grow and produce the plantlets. So you will get tissue culture, or through micropropagation you get lots of, every cell will produce a plantlet, small plant, and then you separate and grow the plant and see which one really gets the, or carry is the your desired gene. You never allow to produce the tumor as it happens in the natural condition. In natural condition, our right hand side image, you can see a big tumor. In that case, thousands of bacteria, millions of bacteria infected this host plant, and over the process, these symptoms appear in the standing plant. But we use the efficiency of the agrobacterium tumefaciens just to transfer the desired gene into the targeted plant cell, not at the plant level. It is at cell level, and then you grow the plant. Okay.
Sir, is there a duration for how
Yes,
long
it
we're gonna
depends. Yeah,
like
yeah,
for the floral
very
dipping?
good.
Like how we are going.
Floral dipping, it is a short exposure, several hours or even a few hours. And after that, uh, it depends on which genotype you are using. So it is not more than several hours or like that. Okay.
Sir,
So
I
like
have a
Yes, Mahmuda, please go on.
one more follow-up question. Sir, is there not work within that time frame because we are just like assuming in the uh, that it might work. It has a certain bit of uncertainty that the plasmid may get inside or may not get inside.
There are huge uncertainty, yes, because what you are doing, you are not cultivating a single cell. Millions of cells of bacteria, millions of plant cells. So some of them might be successful. Okay. Uh, it is not so precise that you use one bacteria, um, uh, one cell of bacterium and one cell of plant and get into the touch. You allow, millions of cells in that case. So through tissue culture, you get, for example, thousands of plantlets, and then check some of them will be successful. And it depends on the protocol. If you uh, can optimize the protocol, even several 30%, 20%, success you can get. That means the plants, uh, transformed plants or modified plants may be higher. And it also depends on the genotype, because in tissue culture, all plants are not equally responding to regenerate into the micropropagation or like that. So it is not 100% precise. You have to screen out the right plant. And once you get a single plant, which, stably modified with your desired gene, that's enough. Then you can make millions, billions copies. Have you got the point?
Yes, sir. Understood.
Thank you.
Thank you, sir.
Thank you. Thank you so much. Any other student, do you have any other question? This slide is very, very crucial and very important and very simplified version of developing transgenic plant. So it may be included in your exam also. Okay. If you have,
sir,
yes.
sir, for the gene gun, is there also like any protocol of how much of like how many particles like we're gonna fire or how much tissue
Yes.
we're gonna use? Is there
Yes,
like
yes.
anything,
There are
any
detailed.
like generalized?
Yes, please continue your
Oh no,
question.
that was my question.
Okay. You mean this is the most simplified illustration. How does it works? But obviously not these three steps. Uh, this is a long procedure and optimized procedure for each plant, uh, how you can get the protoplast, and how you can replicate the gene, and how you uh, you know uh, coat the DNA with gold particles. So it is a long procedure, several steps, and people optimize through the trial and error. But I'm showing here the main principle, because principle is fundamental and very important, rather than the detailed protocol. Detailed protocol uh, depends on case by case, which plant you want to modify, which gene you want to insert into the genome of which plant or like that. So The detailed protocol is not so short, but to understand the principle, I think this slide can serve the purpose. You are right. The detailed protocol is long and it is not general for each plant, uh, for each species, even within the species, each genotype of the plant or variety of the plant has different, protocols. So there are lots of protocol books already been published. For example, you want to modify the genome of maize, wheat, or even potato, there are specific, brinjal specific protocols, but not sure the published protocol can be used in your plant or the gene you targeted, but you need to optimize and need patient work so that finally you will get the success. Once you know the principle, then you can make minute change and improve the protocol to get the higher efficiency. Okay.
Okay. Thank you.
Okay. Thank you. Then I can move other step. Here is very interesting. Uh, for example, you want to uh, how to say, you want to develop a transgenic plant. I previously mentioned that you need to use a specific gene, transgene, gene of interest. So gene of interest, usually we do not use a single gene, that is only the pure gene. No, you need to put two supporter. One is promoter sequence before the gene sequence, and then terminator a sequence. So it is called gene construct and also antibiotic resistant genes. So you need to make a construct to paste into the Agrobacterium tumefaciens and not a single pure gene, because otherwise this gene may be inserted but cannot express and produce the protein. In that case, promoter and terminator and the antibiotic resistant gene, you make a cassette like, construct. And designing and making the gene construct is also very, very crucial. And there are lots of, uh, books, literatures, how can you make it. But the main point is the gene of interest for making the GMO, you must make a construct with promoter of the gene and terminator sequence after the DNA sequence, and then add a gene uh, for antibiotic resistance. And it is called designing and making the gene construct. This is the first step. Second, step is introducing transgene to plants using agrobacterium, as I mentioned before. So agrobacterium you can get from the commercial company. Basically, the wild isolate, they are being commercialized, which are efficient. All agrobacterium are not efficient, which are efficient in transferring gene from its DNA to the plant DNA, and you can use the biolistic gene gun as alternative method. And then third step is growing the transgene plantlets. You grow the plants through the tissue culture and micropropagation. And confirming, fourth step is confirming the plants are transgenic. How can you do it? Through PCR, through phenotypic screening, or gene sequencing. So what transgenics can tell us? Transgenics will give you the additional trait in the GM plant. And this is the called plant genetic engineering. Here is the schematic a representation of agrobacterium-mediated genetic transformation, how it does. Here, agrobacterium cell, and it has, complex TI plasmid, and then you can see this agrobacterium can enter, transfer the DNA, which can virulent gene can bind and enter into the nucleus and can insert the DNA. It is a very, delicate process. You can find several video also, because agrobacterium uh, causing crown gall disease hugely been studied. Molecular mechanism is known, and lots of images you can find. So my interest is not to make this topic more complex, but if you know, due to the presence of TI plasmid and tDNA, it can transfer the DNA into the host or target plant DNA. So there are several factors involved in development of transgenic plants that are agrobacterium mediated gene transfer in plants is highly efficient transformation process, which is governed by various factors including genotype of the host plant, explant, vector, plasmid, bacterial strain, composition of culture medium, tissue damage, or temperature of co-cultivation. So considering all these factors, a protocol is optimized through trial and error. What are the importance of transgenic plants? Transgenic plants are practically very important, especially insect-resistant plant. Insects are uh, uh, destructive for crop cultivation. Many insects damage our crop. For example, I mentioned brinjal shoot and fruit borer. So scientists discovered some bacterium, Bacillus thuringiensis, BT, which produce the toxin for the insect. And it was discovered 100 years ago or more. And people use these bacterial cells directly as biopesticide. But later, when genetic engineering was developed, then no need to use the live bacteria because it has so other problem, living cell, shelf life problem, efficiency problem. So scientists discovered all the genes. The genes cluster is called cry gene. So cry1, SE, or other gene, they can produce express in the plant cell and produce the toxin. So this toxin gene is delivered directly to the plant genome using recombinant DNA techniques, as I mentioned, in case of agrobacterium mediated transformation, and you can protect the plant from the insect pest. Especially BT technology cannot stop all the insect pests, mostly the lepidopteran insects, that is under the order of Lepidoptera. Most of the larvae that damage the crops, they are under that order of the insects. And not only insect resistance, lots of effort has been given to develop virus resistant, plant. Virus not only hazardous for human health during COVID-19 pandemic. We suffered from the SARS-CoV-2 virus, deadly virus, and many people died of COVID-19. Similarly, virus, as they cannot make their own copy, virus particle infect different plants, microorganisms also. So virus can damage tomato, tobacco, papaya, and potato. So, to develop virus resistant plants, transgenic technology uh, has hugely been used. For example, potato virus resistant plant, rice that is resistant to RSV, another virus, and black gram that is resistant to yellow mosaic virus, green gram that is resistant to yellow mosaic virus, and papaya. Papaya mosaic virus is a dangerous virus. In Hawaii, in the US, they were very famous. That island is very famous for, papaya production. Very delicious papaya, famous in the world. But their, papaya industry export was seriously damaged due to papaya mosaic virus. By using genetic engineering, the Hawaii and farmers, protected their papaya production by developing transgenic papaya. So another important use of transgenic plant is herbicidal resistance. Herbicide, what does it mean? We can use herbicide means that kills the herb. When you grow plant, lots of weeds, undesired plant, they grow and compete with your plant. They are called, weed. And weed manually in our country, people used to, clean from their field. But nowadays, many chemical substances targeting the weeds, they are being popular. For example, glyphosate. But in that case, those, herbicide can also affect the plant, your target plant that you are cultivating. In that case, if you make your plant uh, resistant to the herbicide, then your plant will withstand, for example, tomato, potato, tobacco, cotton, and only the herb will be killed. And this is really amazing. And the soybean we are, consuming in Dhaka, most of them are imported, and they are GMO, herbicide tolerant. They are produced from herbicide tolerant or resistant soybean. So this is another huge benefit, and yield can be increased, and also human labor can be saved. To create tobacco plant resistant to E. coli, sulfonylurea, the mutant ALS acetolactate synthase gene from Arabidopsis is transformed to create atrazine resistant transgenic plants. QB protein of photosystem 2, quinone B protein, from mutant amaranthus hybrid is introduced into tobacco and other plants. So lots of techniques to develop the targeted herbicide resistant plant being employed and hugely being cultivated, especially in USA or Canada and many other countries. Nutritional benefits. Every year, 500,000 kids lose their vision completely or partially due to vitamin A deficiency. For a sizable portion of the world's population, milled rice serves as their main food. In that case, golden rice, that is adding vitamin A into the regular major food, is thought one of the approach. So lots of GM crops, genetically engineered transgenic crops, mostly BT or herbicide tolerance, are cultivated worldwide, and they are increasing. Um, so uh, these are the main data. So what are the major steps of engineering a plant? First step is the DNA extraction. You have to extract the DNA from the plant which is resistant. For example, salinity tolerant plant from the Sundarban. You can extract the DNA, and then by analyzing, you can get the gene responsible for salinity tolerant, clone it, and then characterize it. So you have the gene now in your hand. And then you can make copy of the gene by using PCR or other method. Finally, modify the gene by using, promoter, terminator, and antibiotic resistant gene, and then putting the new gene into the plant cell gene gun or agrobacterium mediated transformation. And step 5 is breeding transgenic plant with an elite line. Sometimes, if you develop the transgenic plant, it may not be highly productive, although it is resistant against your target pest, biotic, and abiotic stress. In that case, you need to transfer the gene by crossing and back crossing with the elite variety available in your country. For example, BT brinjal, when gene was successfully transferred from Bacillus thuringiensis to the brinjal genome, then that plant may not readily be commercialized if it is not, uh, you know, efficient in high yield or other, aspects. In that case, gene is now in the brinjal transgene. So what you can do using classical crossing, you can transfer into the high yielding elite variety, and then through back crossing, you can remove undesirable traits and get the gene into your elite variety. And this is the way people are making the new variety of GM crops. Do you have any question? I would like to take a pause here for your question.
Uh, Sir, in the slides for importance of transgenic plant for the virus resistant ones, can you go to the slide?
Yes.
Yes, this first sentence, by adding viral coat proteins, like what exactly is this meaning? Like we're gonna add virus coat proteins to the plants?
Oh, yeah. How virus resistant plants are developed. There are, several approaches. You can use a specific, protein, code protein you mentioned, yes, that you can also develop the transgenic, or you can, make the plant so that uh, plant can easily recognize the virus and activates its immune system so that virus cannot uh, infect and make its own copy. So there are lots of strategies to do that.
Okay. Thank you, sir.
Okay, welcome. And any other question? Thank you for asking the question. And any other student? Any question? No? Then I shall move major steps of transferring a gene from cinnamon plant to apple. You want to, that means in transgenic process, it is quite different than the classical breeding. There is no barrier of compatibility. That means gene of the source of gene can be any plant, any microorganism, any organism. But in case of classical breeding, you need to cross. You can do the cross between two compatible plants. For example, mango and lychee, you cannot make cross. Only mango, different varieties of mango, you can make cross. Uh, Similarly, lychee. So intergeneric crossing is impossible. That means there are some compatibility issues. But in case of genetic engineering, you can take a gene from cinnamon and get the gene and design the construct and then transform the plant And even you can get the cinnamon, principle into the apple. So apple will carry the some desired traits from the cinnamon, and then you have seen here back crossing breeding. Or that means when you get the first transgenic plant, it may not be commercially, profitable. In that, but it carries the foreign gene, transgene. In that case, you can easily cross with the uh, locally elite variety, that is most high yielding and commercial variety, so that gene can be transferred from transgenic to the normal elite variety. Within few years of backcrossing, you can finally get high yielding plant or high quality plant, highly profitable plant with your transgene. So these are the same steps I am showing you can go through. So DNA and DNA extraction, how can you do it? I'm giving you these slides for your reading because DNA is nothing but ATGC, without any full stop. So you can extract the DNA. There are the reagent and kits available to extract the DNA from the plant cell. Lots of methods are optimized. It is not a tough job. And then how can you get the gene from the DNA? The genetic language could be compared to our language DNA. I mean, there are 26 letters in English alphabet, and in order of those letters makes up different words with unique meaning. But in case of DNA, only four letters. The whole book is written into four letters, but telling us lots of stories. For example, in human DNA, 3.2 billion base pairs. For example, this is a base pair AT. So 3.2 billion base pairs, but all the base pairs are uh, uh, you know mainly are composed of four letters at EGC, and they make how to say 30 to 40,000 genes, and the huge complex Homo sapiens, and 8.4 billion people on this earth at this moment. They are unique due to the sequence variation. So in the DNA, gene 1, gene 2, gene 3, sometimes within the gene there might be non-coding region. So there are lots of computational analysis you can and genetic analysis, molecular analysis. How can you map a gene and clone a gene? So after mapping and then cloning, finally you get a gene. And then once you clone it, then the DNA makes up the genes is the same DNA. These are the small. How can you clone? Clone means you figure out the sequence of the gene and then synthesize that gene and integrate into a bacteria. Bacterial, transformation is very easy. As I mentioned before, you can easily paste into the plasmid. And if you see that gene express, for example, a gene responsible for blue fluorescence. If you see bacteria is emitting blue fluorescence after integrating the gene, that means you are perfectly, got the gene. Once you characterize and clone the gene, and then uh, you can easily insert it into the DNA, and there are lots of process. Please go through how people discover a gene and then modify the gene. As I mentioned, you asked the protocol how a gene can be modified. Here you see promoter, terminator sequence, and make the construct. And then finally, you uh, um, use the transformation and how agrobacteria mediated transformation. Here I have seen this is agrobacteria, TI plasmid. You made the TI plasmid cut into the restriction enzyme and put into your DNA, and finally how transformation taken place. Here is the pathway I showed. So please go through for the reading materials. In the next class, face to face, I shall allow you to, ask the question. We can not only use the plant cell, even pollen or anther can be used for transformation and get the plant. Even as different kinds. Here is the detailed procedure. You can go through how many hours in which condition we can do. Here is another one. General steps in agrobacterium mediated transformation of cereals. Cereals means rice, maize, or wheat. All the steps in detail. So you please go through as you know the principle. And transgenic plants, when you got, carries the foreign gene, for example, BT gene, but it is dwarf, less productive than the elite line. In that case, you can transfer the gene into this plant through the crossing. So you take the pollen from this one and cross this one, and you will get the hybrid, and then you see whether gene is transferred by PCR. You can check, and uh, by several how to say back crossing, you can get the elite line carries with newest elite line. Here you can see high yield and also carries your gene that is interesting to you. So this is all about today's lecture, and I have shared the PDF file with you, so you can go through. And there are lots of videos also on genetic transformation, even uh, the detailed images, because genetic transformation of plants, that is GMO or transgenic, we have more than 40 years of experiences. So lots of data generates and lots of issues are associated with genetic transformation. I shall discuss later on the biosafety issues associated with genetic transformation. So do you have any question? You can ask some question, and then I shall stop today's lecture.
Sir?
Yes.
Sir, this agrobacterium method and the gene gun method, we will use these same methods for even like making cisgenic plants, right? You mentioned cisgenic plants last time.
Cisgenic, yes. Cisgenic plant, that is if you take the gene from rice, for example, aromatic rice, to introduce into non-aromatic rice. Yes. If a gene is cloned, uh, source is no matter. You can handle in the same
Yeah.
way.
Okay.
Thank you. Any other?
Sir, in the
Yes.
next class, we are going to have a quiz, right?
Yes. Actually, due to several reality, that is reality. Today I had a plan to take the quiz. So next class, uh, uh, will it be online or in person? Have you
Sir,
checked?
I think
In
we're going to
person,
have
sir.
Hmm?
a in-person class, sir.
Next class
Offline
will
class.
be 21st February.
Oh, that's a government holiday, sir.
Yes. So in that case, if I told you in earlier class that can you communicate with the department administration whether they can keep a classroom open on Friday, then we can take the quiz exam for an hour only is okay. So if you can contact with the department administration and arrange a room, then you can have a quiz on 20th of February instead of 21st. Is it possible? Can anyone volunteer? Can anyone volunteer? Hello? If not, then maybe next Friday, we shall have another online lecture, and it would be hard for you to carry on the, quiz exam.
স্যার আমাদের কি অনলাইনে কুইজ নেওয়া পসিবল হবে?
No, অনলাইনে কুইজ নেওয়া নানা রকমের সমস্যা হবে।
স্যার আসলে অনেকগুলো ক্লাসের পড়া একসাথে কুইজে অনেক প্রেসার পড়ে যাবে
এই
আমাদের।
জন্য বেস্ট আইডিয়া হচ্ছে তোমাদের 2-3 জন একটু অ্যাডমিনের সাথে কথা বলুন, শুক্রবারে 1 আওয়ার বা 1 আওয়ার 30 মিনিটস ফ্রি রাখো আমাদের জন্য একটা সেশন, যে কোনো একটা ক্লাসরুম হলেই হবে। তাদের সাথে একটু কমিউনিকেট করো যে আমাদের অনেকগুলো
I don't know who is the admin here. স্যার অ্যাডমিন কে বা সিআর?
আমি এই যে মুস্তাক সাহেব আছে অথবা আরেকজন ফিমেল, তুমি ডিপার্টমেন্টে জিজ্ঞেস করলেই হবে, ডিপার্টমেন্টের হেডের সাথেও কথা বলতে পারো, দ্যাট উড বি ফাইন, চেয়ারম্যান ইফতেখার সাহেবের সাথে। ঠিক আছে, তোমরা 2-3 জন গিয়ে বলো যে এরকম আমাদের তো একটা রিয়েল প্রবলেম তৈরি হয়েছে।
স্যার আমি মেল দিতে পারবো, বিকজ আমি নিজে আর কি হোম টাউনে আছি, তো এর জন্য আর কি।
না, যে কোনো একজন, এখানে তো সবাই তো আর হোম টাউনে নেই, তাই না?
জি স্যার।
এই সপ্তাহের ভিতরে আলাপ করে এটা আমাকে জানালে তখন মনে করো আমরা ইন পার্সন একটা কুইজ নিয়ে নেওয়া বেটার। ঠিক আছে, আর কে দায়িত্ব নিতে পারবা?
স্যার আমাদের সিআর আছে কি আমাদের কোর্সে?
আমি জানি না তো।
স্যার কোনো সিআর থাকলে উনাকে বলে যদি করানো যেত আর কি, তাহলে বেটার হতো।
তোমরা তোমরা চেয়ারম্যানের সাথে 2-3 জন স্টুডেন্ট ভলান্টিয়ারলি যাও, গিয়ে কথা বলো। ঠিক আছে, এবং আমাকে জানাতে পারো যে স্যার অমুক বা ওনারকে ইমেইল করলে হবে বা যে কোনো ভাবে। ঠিক আছে, আমি কি বোঝাতে পেরেছি? কি বলো তোমাদের দিক থেকে? আর কোনো প্রশ্ন আছে কিনা তোমাদের?
স্যার
একটা
যদি কুইজ
বলো জান্নাতুল ফিরদাউস।
স্যার যদি ফ্রাইডেতে কুইজ হয় স্যার, তাহলে স্যার সিলেবাসটা কতটুকু থাকবে স্যার?
সিলেবাসটা আমরা সেট করতে পারবো। তোমাদের ক্লাস অনেকগুলো হয়েছে, তো আমরা মনে করো টিস্যু কালচার পর্যন্ত একটা কুইজ নিয়ে নিলাম। ঠিক আছে, অথবা
লেকচার 1 আর 2 টা
না, লেকচার 1-2 এবং আজকেরটা তো 3-4, কাজে 1-2-র ভিতরে একটা কুইজ নিয়ে নিলাম। তোমাদের ফার্স্ট কুইজটা একটু রিল্যাক্স হলো, অথবা 1-2 এবং আজকেরটাও ইনক্লুড করা যেতে পারে, কিন্তু যত দেরি হবে তত তোমার লোড বেড়ে যাবে। দেখো আলাপ করে। ঠিক আছে, এইটুকুই থাকলো। আর কারো কোনো প্রশ্ন আছে? নেই? থ্যাংক ইউ ভেরি মাচ।
স্যার আমার একটু
বলো।
একটু ওই প্ল্যান্ট টিস্যু কালচারের ওখানের মধ্যে আর কি, যে আপনি যে মাইক্রো প্রপাগেশন পড়িয়েছিলেন, সো মাইক্রো প্রপাগেশন করার জন্য তো ওয়ান অফ দ্য মেইন স্টেপ হচ্ছে কি আমরা টিস্যু কালচার আগে করব, রাইট?
ইয়েস, টিস্যু কালচারের মাধ্যমে প্লান্ট যখন আমরা কমার্শিয়ালি কালটিভেট করি, এবং একেবারে টিস্যু কালচার তো টিস্যুকে কালচার করছো, কিন্তু টিস্যু কালচার করে যখন একেবারে তুমি সিডলিং পাচ্ছ, এই সিডলিংকে রিয়েল ফিল্ডে কালটিভেট করছো, এইটাকে বলে মাইক্রো প্রোপাগেশন। একেবারে তুমি টিস্যু সেল থেকে শুরু করে তুমি বীজ এখানে ইউজ করছো না, তাই না? আমরা যেমন ধান চাষ করার জন্য, গম চাষ করার জন্য, আলু চাষ করার জন্য বীজ ইউজ করি, কিন্তু এখানে টিস্যু থেকে কালচার করে আমরা চারা পেয়েছি, এই চারাকে আমরা ফিল্ডে গ্রো করে কমার্শিয়াল কাল্টিভেশন করছি, এটাকে মাইক্রো প্রোপাগেশন বলে। কাজেই মাইক্রো প্রোপাগেশন করতে হলে টিস্যু কালচার মাস্ট, ঠিক আছে? আর এই মাইক্রো প্রোপাগেশন আর টিস্যু কালচারের পার্থক্যটা খুব ক্লিয়ারলি বুঝতে হবে। টিস্যু কালচার মানেই মাইক্রো প্রোপাগেশন না, তাই না? কাল্টিভেশন অফ প্লান্ট টিস্যু or cell into the culture media in aseptic condition we can call the tissue culture. If it goes up to the field cultivation through the seedling developed through tissue culture is called micropropagation. And tissue culture এর চারাকে ডাইরেক্ট ফিল্ডে নেওয়া যায় না, তোমাদের এটা বলছি কিনা। এই চারাকে প্রথম তোমার অ্যাসেপ্টিক কন্ডিশন থেকে বের করে হার্ডেনিং করতে হয়। হার্ডেনিং মানে ওই সিডলিং গুলো খুবই দুর্বল থাকে নরমাল পরিবেশে যাওয়ার ক্ষেত্রে, এইগুলোকে টবের মধ্যে লাগিয়ে ছায়াযুক্ত জায়গায় পানি টানি দিয়ে আস্তে আস্তে নরমাল তোমার এনভায়রনমেন্টে নিতে হয়। যেমন মনে করো, দেখবা যে এই অপারেশন করার পরে এরপরে একটা স্পেশাল কেয়ার ইউনিটে রাখে, এরপরে বেডে দেয়, তাই না? তো এইরকম একটা অবস্থা কি হার্ডেনিং। হার্ডেনিং হচ্ছে একটা প্রসেস যেখানে প্লান্টটাকে নরমাল এনভায়রনমেন্টে নেয়ার জন্য তাকে ফিট করা হয় একটা ইন্টেন্সিভ কেয়ারের মাধ্যমে।
ওকে স্যার, আচ্ছা লাইক তো যখন আমরা মাইক্রো প্রোপাগেশন করব, ওটার জন্য কি মানে এরকম স্পেসিফিক কয়েকটা টাইপ অফ টিস্যু কালচার আছে? যেরকম আপনি লাইক ইয়ার মধ্যে মেনশন করেছেন মেরিস্টেম দিয়ে আমরা কালচার করি, এমব্রয় দিয়ে হয়তো কালচার করা হয়, ওটার উপর শুট কালচার করা হয়, এগুলা কি মানে স্পেসিফিকলি মাইক্রো?
না, মাইক্রো প্রোপাগেশনের ক্ষেত্রে যেহেতু এটা কমার্শিয়ালি করা হয়, নানা রকম চেষ্টা করে কোনো একটাকে অপটিমাইজ করে ফেলেছে যেটাতে তোমার কস্ট কম, এফিসিয়েন্সি হাই, হ্যাঁ, এবং কম স্টেপ লাগবে, এগুলোকে অপটিমাইজ করে নেওয়া হয়। কারণ এটা যেহেতু কমার্শিয়ালি লো কস্ট হইতে হবে, খুব হাই নাম্বার অফ তুমি সিডলিং পেতে হবে, তাই না? এটা তো রিসার্চ না, এটা কমার্শিয়াল, সেই জন্য এটাকে একেবারে অপটিমাইজ করা হয়। যেমন তুমি ব্রাকে যদি যাও, ব্রাক টিস্যু কালচারে চারা কিনতে, হ্যাঁ, তারা এখানে এক্সপেরিমেন্ট না, এরা মনে করো স্ট্রবেরির চারা হোক অথবা স্টেভিয়ার চারা হোক অথবা বানানার চারা হোক, হ্যাঁ, এইগুলো তারা অপটিমাইজ করে ফেলেছে এবং রুটিনলি একই মেথডে ইউজ করছে, এখানে এক্সপেরিমেন্ট তারা করে না। ইভেন পটেটোর সিডলিং যদি তুমি তারা কোন অংশটা থেকে করবে, কোনটা থেকে করলে মোস্ট এফিশিয়েন্ট হবে, হ্যাঁ, কম স্টেপ লাগবে, প্রফিটেবল হবে, কম এনার্জি লাগবে, কম মিডিয়া লাগবে, সব কিছুকে অপটিমাইজ করে ফেলে। হ্যাঁ, টেম্পারেচার থেকে শুরু করে মিডিয়া কম্পোজিশন এবং সবকিছু বিষয়, ইভেন কোন কন্টেইনারে করবে তাও তুমি অপটিমাইজ করে ফেলে, কারণ এখানে ইকোনমি একটা বিরাট ফ্যাক্টর। ইকোনমিক্যালি ইট মাস্ট বি প্রফিটেবল।
তো স্যার, তো যখন আমরা বলি কি মাইক্রো প্রোপাগেশন আর টিস্যু কালচারের মাঝখানে ডিফারেন্স কি? সো যে কালচার মিডিয়া কালচার টাইপটা ইউজ করা হবে ওটাও তো একটা ডিফারেন্সের আন্ডারে পড়বে, রাইট?
ইয়েস, ইয়েস। টিস্যু কালচার তুমি যে কোনো মেরিস্টেমেটিক টিস্যুকে নিয়ে করতে পারো, এবং এটা যদি মাইক্রো প্রোপাগেশনে যাও, তুমি দেখবা যে কোন এই ইয়ে থেকে, অর্গান থেকে, কোন মেরিস্টেমেটিক টিস্যু থেকে, যেমন পটেটোর ক্ষেত্রে মনে করো যে টিপ ইউজ করছে, হ্যাঁ, অথবা টিউবার ইউজ করছে, কোনটা ইউজ করলে তুমি মানে কমার্শিয়ালি প্রফিটেবল তোমার আউটপুট পাবা। এইটা হচ্ছে তোমার মাইক্রো প্রোপাগেশনের একটা বড় প্রিন্সিপাল। তুমি অবভিয়াসলি টিস্যু কালচার করবা, কিন্তু কোন অর্গান থেকে করবা, হ্যাঁ, এটা ডিপেন্ড করে কোনটা থেকে হায়ার এফিসিয়েন্সি, হায়ার নাম্বার অফ প্লান্টলেট তুমি পাচ্ছ এবং ফাস্টার পাচ্ছ। থ্যাংক ইউ, ভালো প্রশ্ন করেছো।
স্যার, আরেকটা কোয়েশ্চেন, এটা মেবি এত ইম্পর্টেন্ট না, আমি জাস্ট আনসারটা খুঁজে পাচ্ছি না থেকে ভাবছি আপনাকে একটু জিজ্ঞেস করে নেই। যখন আমরা ওই হিস্ট্রি অফ প্ল্যান্ট টিস্যু কালচারের মধ্যে আপনি একটা মেনশন করেছিলাম একটা আহ, একটা হাইব্রিড তৈরি করেছিল, যে হাইব্রিডটা আমার লাগে তৈরি করে হয়তো ওরা ক্যালাস প্রথমবার যখন করলো, যে ক্যালাস কালচারটা যেখানে করেছিল নিকোটিনা গ্লাইকা নাইন নিকোটিনা ল্যান্সডরফিল এরকম একটা হাইব্রিড ওরা নিয়ে কাজ করেছিল। ওটা নিয়ে আমার লাগে এখনো কাজ হচ্ছে, লাইক ওরা হাইব্রিড স্টাডিজ আর কি ওটার মধ্যে করে, বাট আমি না বুঝতে পারতেছি না এটা ওরা প্রথমবার বানিয়েছিল কেন, কোনো
হ্যাঁ,
পারপাস ছিল নাকি এটা।
যেমন আমরা হাইব্রিডাইজেশন করি কি ফ্লাওয়ারের পলিনেশনের মাধ্যমে, তাই না?
হুম
কিন্তু
হুম
তোমার টিস্যু কালচারেও হাইব্রিডাইজেশন করা যায়। মনে করো দুটো
হুম
আহ, এই প্ল্যান্টের একই স্পেসিসের
হুম
কিন্তু দুই রকম, একটা রোজের কালার হচ্ছে এই পিঙ্ক, আর একটা মনে করো যে কালার হচ্ছে রেড, অথবা একটা হচ্ছে হোয়াইট আর একটা হচ্ছে পিঙ্ক, দুটোকে ক্রসিং করে তুমি নতুন কালার চাও। তো সেই ক্ষেত্রে এই সেলকে ফিউজ করা হয়, এটাকে প্রোটোপ্লাস্ট ফিউশন বলে। হ্যাঁ, এই দুটো সেলকে
হুম
ফিউজ করে বাই ফোর্স তুমি হাইব্রিডাইজ করে ফেলতে পারো। এবং একটা সিঙ্গেল সেলে প্রোটোপ্লাস্ট ফিউশন এই ক্ষেত্রে সেল ওয়ালটাকে রিমুভ করে তোমার নানাভাবে এই প্রোটোপ্লাস্ট ফিউশন করার প্রসেস আছে। এরপরে সেলটাকে কালচার করে যখন প্লান্ট পাবা, প্ল্যান্টের মধ্যে তুমি হাইব্রিড প্লান্ট পেতে পারবা, এবং এই ক্ষেত্রে দুটোর জেনেটিক রিকম্বিনেশন হয়। কাজেই প্রোটোপ্লাস্ট ফিউশন করে অনেক সময় খুব ইন্টারেস্টিং হয়, যেমন পটেটো এবং টমেটো। হ্যাঁ, পটেটো টমেটোকে ফিউজ করে পমেটো পেয়েছে। মানে এটাতে এই কাণ্ডের মধ্যে তোমার টমেটো ধরছে, আর এই শিকড়ের ওখানে তোমার পটেটো ধরছে। কাজে নানা রকমের এক্সপেরিমেন্ট এরকম আছে, সায়েন্টিস্টরা করে। টিস্যু কালচার একটা বেসিক টুলস, যেখানে সেল লেভেলে, মলিকুল লেভেলে, জেনেটিক্স লেভেলে প্রচুর স্টাডি করা যাচ্ছে। মানে এক্সটার্নাল ইন্টারফেয়ারেন্সের বাইরে, যেহেতু এটা একটা কন্ট্রোল এনভায়রনমেন্টে করা যায়, কাজেই দেখা হয় দুয়ে দুয়ে চার হয় কিনা, অর্থাৎ জিনগুলো যখন তোমার রিকম্বিনেশন করে, অথবা সেল বায়োলজিকে বিভিন্ন ফ্যাক্টর দ্বারা যে নিয়ন্ত্রিত হয়, এগুলো এই টিস্যু সেল এবং টিস্যু লেভেলে কাজ করতে গিয়ে আহ, খুব ইজি। এবং অনেক সময় সেলের ভিতরে কিভাবে মলিকুলার ফাংশন হয়, এটা ইন্ডিভিজুয়াল সেলকে প্রোটোপ্লাস্ট তৈরি করে তুমি মাইক্রোস্কোপে দেখতে পারো, ভিজুয়ালাইজ করে, প্রোটিনকে ভিজুয়ালাইজ করে বা অর্গানেলিকে ভিজুয়ালাইজ করে। কাজে এই টিস্যু কালচার অনেক পাওয়ারফুল টুল তোমার বেসিক রিসার্চের ক্ষেত্রে। জান্নাতুল ফেরদাউস, তুমি হাত উঠিয়ে রাখছো,
থ্যাংক ইউ
জি
স্যার।
স্যার। স্যার
বল।
আপনি বললেন যে হচ্ছে মানে স্যার আমাদের ক্লাসে অনেকেই স্যার টিআর এ আছে স্যার, ওরা চাইলেই স্যার খুব ইজিলি স্যার অফিসে কন্ট্যাক্ট করতে পারে স্যার, এই জন্য হাত উঠিয়েছিলাম
এইটা
আর কি।
এইটা বিগ জব না, গিয়ে একটু আলাপ করো। আলাপ করে আমি মনে করি যে তোমরা এটা সেট করতে পারো। এটা নিজেরা কিছু ভলান্টিয়ারলি জব করতে হয়, এবং এটা খুব কঠিন জব না। যদি শুক্রবারে তাদের প্রিন্সিপাল থাকে যে এটা সম্ভব, দেন আমরা খুব কুইক ডিসিশন নিব। এই ফেসবুকে জানাই দিবা, তখন রুম নাম্বারটা জানিয়ে দিবা। আমার যদি রিকুইজিশন দিতে হয়, হ্যাঁ, যদি আমার কিছু লাগে, তখন ফর্ম নিয়ে আমাকে পাঠিয়ে আমি ই সিগনেচার যোগ করে দিয়ে দিয়ে দিব। ঠিক আছে, থ্যাংক ইউ। তোমরা তোমাদের বন্ধুদেরকে জানো যারা ওখানে টিএ বা হিসাবে আছে, তোমরা যে কোনো একজন গেলেও হবে। ঠিক আছে, আমরা খুব ভালো হয় শুক্রবারে একটা তোমাদের কুইজ নিতে পারলে। কারো কোনো প্রশ্ন আছে আর? আমি কোনো রেইজড হ্যান্ড দেখছি না আর। যদি না থাকে আমি এখানে আজকে শেষ করব। তোমাদেরকে অসংখ্য ধন্যবাদ। এবং আমি এখন ময়মনসিংহ কৃষি বিশ্ববিদ্যালয়ের ব্রহ্মপুত্র নদীর পাড়ে একটা সুন্দর গেস্ট হাউস আছে, হ্যাঁ, এখান থেকে তোমাদের সাথে কথা বলছি। কালকে একটা ইন্টারন্যাশনাল কনফারেন্স আছে, কনফারেন্স করে জাপানিজরা অনেকে আসছে, কালকেই ব্যাক
স্যার
করব।
আমিও ময়মনসিং থাকি।
ও গ্রেট,
আমি
তাহলে
ওই ময়মনসিং ওই দিকেই থাকি, মানে টাউনে।
ও গ্রেট, তোমাদের ময়মনসিংহ টাউনে আমাদের যৌবনের প্রচুর সময় কাটিয়েছি। বাংলাদেশ কৃষি বিশ্ববিদ্যালয়ের লম্বা সময়, 85 থেকে 92 পর্যন্ত, এখানে অনেক লম্বা সময় 91-এ আমাদের আন্ডারগ্রেড হয়। এরশাদ সাহেব তখন ক্ষমতায় ছিল, ইউনিভার্সিটি প্রায় বন্ধ হয়ে যেত, অর্থাৎ 4 বছরের আন্ডারগ্রেড আমরা 7 বছরে কমপ্লিট করেছি। এটা তোমাদের জেনারেশন ইমাজিন করতে পারবা না আসলে কত ডার্ক ওয়ার্ল্ডে আমরা ছিলাম। কথাবার্তা নেই, ইউনিভার্সিটি তোমার 6 মাস 9 মাসের জন্য বন্ধ হয়ে যেত, কারণ সামরিক আইন বা অন্যান্য। এখন আমরা এই দিক থেকে কিছুটা ভালো আছি এবং আমরা ভালোর দিকে যাচ্ছি আশা করি। তো সবাইকে এই ভালোবাসা দিবস, সমস্ত মানুষের প্রতি আমাদের ভালোবাসা থাকলো, এবং সবাই সামনে রামাদান আসতেছে, সবার জন্য অনেক শুভকামনা। আর নতুন সরকার এসেছে, সরকারের জন্য শুভকামনা। আমরা আমাদের দেশটাকে খুব সুন্দর দেখতে চাই, আর তোমাদের প্রত্যেকের জন্য শুভকামনা রইল। গুড নাইট।
স্যার, স্যার একটা স্যার, আমি আমার অ্যাটেন্ডেন্স লিস্টে আমার নামটা ছিল না স্যার। স্যার, এটা কি এখন আপডেটেড হয়েছে কিনা এটা একটু দেখার জন্য স্যার।
না, হয়ে যাবে,
রুবাইয়া শামসি।
ঠিক আছে। তুমি আমাকে একটা টেক্সট মেসেজও দিয়ে রাখতে পারো, আর এটা অটোমেটিক তোমার এই অ্যাটেন্ডেন্স রেকর্ড করছে, ঠিক আছে?
ওকে স্যার, থ্যাংক ইউ।
ওকে, হ্যাঁ, থ্যাংক ইউ।